Thank you so much, George, and thank you everyone for being together today. It's an absolute delight to get to moderate this panel. When Drew asked me to do this, my initial reaction was, I get to hang out with many of my scientific heroes for an hour. How can I possibly say no? I literally grew up reading the papers these guys wrote by my bedside. We're going to chat about the Cohort initiative, but before that, I wanted to prepare our keynote speaker this year, those who will be heard from later from the Gates Foundation, just to warn him. Usually you agree to give a keynote speech at some meeting, you think that'll be the end of it. That's not the case here. This initiative has come directly out of the keynote speech that Elias gave exactly 1 year ago at Lausanne VI. And in a remarkable demonstration of how impactful and applied this meeting is, 1 year on, here we are talking about a very specific, substantial initiative to have come directly out of the last meeting. So this really is a remarkably astute example of how applied this meeting is. To start, I'll ask our panelists to briefly introduce themselves, and we'll get into it, not that they need any introduction. Elias, would you mind going first?
Yes, thank you, Nadeem. I'm Elias Zerhouni. I'm actually a professor right now at Johns Hopkins University, my alma mater. And recently I was the head of global R&D for Sanofi. And prior to that, I was at the Gates Foundation as a senior fellow. And prior to that, I was the director of the National Institutes of Health here in the US from 2002 to 2008. And prior to that, I was at Hopkins. I was the Dean for Research and Executive Vice Dean and various other things.
I do feel a bit silly asking you to introduce yourself, Elias. My apologies. And then next, on my screen, I see Andrea.
Yeah, good afternoon, good morning. It's a pleasure to be here. I'm probably one of a few ones actually being in Lausanne. So yeah, I'm the founding CEO of AC Immune, a company working on novel therapies and diagnostics in Alzheimer's. Before that, I was the global head of research of Nestle for more than 20 years. And before that, actually under Elias, I was working at the National Cancer Institute trying to figure out how to link mutations to oncogenes and tumor suppressor genes and actually do precision medicine. So I'm very happy to be here.
Thanks so much. Hakon?
Yeah, good morning. My name is Hakonarson, and I'm a professor of pediatrics at the University of Pennsylvania and Children's Hospital of Philadelphia. I'm a physician scientist. I direct a large-scale genomics operation here at CHOP, and I have extensive collaboration with the Alzheimer's Genomics Consortium. I co-chair the IHC cohort engagement project, and I co-chair the cohort effort here with Dr. Zerhouni.
Great, thanks, Hakon. And Rhoda?
Hi, I'm Rhoda O, and unlike everybody else, I've been like at one place. I've been at Boston University, and I've been a very long-standing investigator for the Framingham Heart Study. Although I am trying to move my identity beyond Framingham, and this Davos initiative is certainly helping me do so. And so I have a lot of interest in other parts of the world, including China in particular.
Great, thank you all. And just for completion, I'm Nadeem Sarwar. I'm the founder and president of the Eisai Center for Genetics Guided Dementia Discovery, or GTD2. Before Cambridge, US, I was in Cambridge, UK, where I was an epidemiologist. Before epidemiology was cool, we got in there first. So to kick things off, like I alluded to before, this is a remarkable session because it came directly out of the last meeting where Elias gave this tremendous keynote lecture about why a new and very focused and applied cohort would be a key next step in helping us solve and eradicate Alzheimer's disease. So to start with, I wanted to invite Elias and Hakon to give an update on what's happened since we last met. There's a lot has happened, and what is the current status of the cohort project. So Hakon and Elias, over to you guys.
Yes, thank you, Nadeem. First and foremost, I'd like to thank George for having us together and to have really motivated us to do something after the last meeting, and invited both Hakon and myself to work on the idea of a global cohort. But to start with, let me give you the background of why we thought it was very important. As we did a very superficial analysis of the research in Alzheimer's, we realized that there was a major gap, and the gap is the fact that no one has a large enough, diverse enough cohort to truly understand the heterogeneity of the disease, its natural history. Because it's a long natural history, it's really important to understand why is it that it comes up in certain patients and not others, how does it come up in certain countries and not others, what are the environmental factors, and so on. So we put together a really fantastic team. I mean, I'm speaking really here for 60 plus experts that have come together to formulate what a global cohort should look like. And as we worked on it, we realized that scientifically it was very important to have that scale. And the reason why scale is important is because as you deal with the long natural history of this disease, you realize that you have to detect it early. When you intervene too late, the damage has been done, and we needed to find ways of identifying the appropriate population, not just for clinical trials, but to understand and have clues as to exactly what are the drivers of this disease as early as possible. Key to that is the example of the Framingham study. I'm so happy to have Rhoda here, because this is the granddaddy of all cohorts that has taught the world about the risk factors in heart disease. Let me give you an example of why it is important to have a scale. Because if you look at the Framingham cohort, it started in 1948, it was 5,000 patients, but the findings became obvious once we followed up these patients for 10 years, 12 years. So if you really look at it, it took about 50,000 years of life years of observation before elements could come up. And so Hakon and I thought about it and said, what do we need to do to get to that scale as rapidly as possible? And this is where we worked with Hakon and the IHCC, which has cohorts around the world that have millions of individuals, but not focused on Alzheimer's disease. And so the idea here is to truly, if I can have the next slide, if you could, is truly diversify the source of knowledge. Right now, 95% is done, of the research is done on Caucasians, and therefore you can't really contrast and compare the genomics, the phenotypes, the biomarkers between different populations to sort of extract the signal from the noise. And this is the fundamental reason why we decided to have a program that would have what we call a trunk. It must have, if you will think of it as a tree, there will be a trunk of data that we need to have that will assemble as many life years of observation as possible right from the get-go, and then enrich that with branches that are going to look at different aspects, if you will, of the disease process, and they hopefully identify in a precision medicine approach and classify, if you will, these individuals. And so we came up with a very clear scientific plan. We are working on what we call the foundational phase, because as you can imagine, doing a global trial of this magnitude has never been done. Rules and ethical rules are different, techniques are different in different countries, access to resources is different. And so we are working at this in this foundational phase on specific pillars of one, accumulating enough follow-on data in existing cohorts. And as you'll see from Hakon, it's doable. And actually at the start, we'll have about 120,000 life years of history, if you will, already at the beginning. And then enrich this cohort with the must-have data that will be harmonized around the world, learn as we do this, and train the individuals and the sites that will participate, and then scale up to the size that is necessary for us to penetrate the secrets of this disease. So I'll turn it over to Hakon at this time, because he really has been the mastermind behind how to construct such an enriched cohort for us. Hakon?
Yeah, thank you, Elias. And maybe if you move to the next slide. So this slide really just shows the pillars that Elias was referring to. And the goal here is to leverage the cohort sort of franchise that we have already assembled as a part of the IHC Consortium, which is now a partner with DAC. And this actually includes over 100 cohorts, each with at least 100,000 individuals or more, And this is a diversity cohort from all over the continent and includes over 60 million individuals today. And what we started doing was to sort of explore what is currently available. And we looked at phenotypic data, risk factors for Alzheimer's disease, cognitive assessment, imaging data, biomarker data, and alike. And we surveyed these cohorts, and we got response from over 30 almost immediately, all keenly interested in participating. And they have now sent us feedback in terms of the information that they have. And if you go to the next slide, the next slide sort of shows the demo, if you will, of what is currently a reference data set for a European ancestry, the status of sort of, Alzheimer's research today. The UK Biobank cohort is probably one of the most complete cohorts that we have, but it's largely European, as you know. And so we took steps to basically look into South America, Africa, Asia, and other countries, Korea, Japan, China, Singapore. And we have now sort of, a list of surveys where we have compiled information, as you can see here on the left graph, in terms of what is available. And the green sort of color here implies that everyone is with those data. So phenotypic data, for example, in the UK, GWAS data, exome data, but there is missing data in some of these cohorts, and that is what we are now going to be gap filling, and what we will start with in this sort of pilot phase, as Elias mentioned, is to get 12,000 individuals with comprehensive phenotypic assessment across the these sort of various elements in terms of having cognitive assessment done in an individual in the age range of 50 to 65. And from the compiled list of surveys that we have over 500,000 individuals who are in the age range where data is available on a subset of individuals. So compiling 12,000 together with some financial support from the DAC consortium, we believe is very manageable over the next few months. And then if you go to the next slide, the goal, and you can sort of see the continents that have already responded and surveyed into the consortium. And if you go to the final slide, this will be sort of gap filling now with, if you advance the slide, please, to the next one. We will be gap filling with supporting data, new data generation across these various domains of imaging, biomarkers, digital biomarkers, and phenotypic data to the extent needed. So we will have additional 6,000 individuals with very comprehensive data to sort of conduct the pilot that we sort of, do as this foundation phase, and will dictate how we move forward with the full 5-year program.
So as you see, we are building not from scratch, we're building from a base that is already existing. And we're really choosing a combination of cohorts, of small cohorts that will form a good landscape, if you will, of the disease around the world. And on average, what we would like to have are individuals in these cohort subjects that have at least 10 years of follow-up. So if you really go into what I count as the key here, life years, we'll have 120,000 to start with, which is what about the Framingham cohort had in terms of the beginning of its ability to extract knowledge out of the Framingham cohort. In this case, we want to innovate as well. And before I end, I would like to really thank Rhoda, because we're going to introduce biomarkers that have never been used before. This is the second pillar that Hakon is talking about, where we'll gap fill to current state of the art, including digital biomarkers, which she will talk about. So with that, I'll turn it back over to you, Nadeem.
Tremendous. Thank you so much, guys. And just a suggestion to George and Drew and the organizers, I think this could become the new bar of if you give a keynote lecture or you agree to give a keynote lecture at a Lausanne meeting, 1 year later, you have to come back with a game-changing proposal like this. So good luck both with that. But this really is an effort to start a new paradigm for Alzheimer's research and allow Alzheimer's to catch up with other areas. Just a couple of follow-up questions, if I may, Elias, initially to you. One thing I've been struck by being involved in this effort is how multidisciplinary the team is that you brought together. This is quite different to how traditional epidemiological or cohort research is done. Usually an academic center of excellence drives the initiative. Once it's established, then new collaborators are added. But this is different. Can you describe a little bit about the multidisciplinary and collaborative approach you're taking?
Right from the beginning, I thought we understood that convergence of multiple strands, if you will be necessary. And that's really George's talent, to bring people together from different walks of life, because this is going to be complex. For example, repositories. So we went to experts who understand repository, sample management, and so on. Digital biomarkers, we went to Rhoda, who's a leader in the field, and we established working groups in the different streams, if you will, the work streams. And at the end of the day, we realized that you cannot do a comprehensive global cohort without engagement and commitment from multiple areas of science. And this is actually the weakness of existing cohorts. They're too narrow and too vertical. And the standards are not applied from one cohort to the other, so that you can never aggregate data. So you realize that there is no choice. You have to be multidisciplinary, and you have to create this sort of ecosystem. And I tell you, I've worked on cohorts and worked on large projects in my career, at NIH in particular. I've never seen people come together the way this has come together. I was quite skeptical when George said, let's go and do it. And he brought people together from different areas of science. Each one of them is contributing a brick to that foundation that at the end of the day will help humanity.
Yeah, I think that's often something that we as scientists need to do a better job of, is communicating the common mission, regardless of sector, whether it's academia, industry, philanthropy, public, private. Ultimately, the reason we wake up in the morning and go to work is to help people live longer, better lives. And initiatives like this really help bring that to the front. That, here's what we're trying to do. We're trying to eradicate Alzheimer's and deliver new medicines. And here's what each of our sectors can bring to the table to make that happen. One specific question, Hakon, for you, that's also my personal bias around genetics and genomics. So like I said, I'm biased here, but I sense that the low-hanging fruit for an effort like this really may reside in the genomic space. Like Elias alluded to, the real value, like a kind of fine wine as it matures in a large cohort study, that you accrue person years of follow-up. But with genomic data, there may be actionable insights you can get very quickly. Would you mind describing a little bit about the kind of genomic and kind of omic aspect of the cohort?
Thank you, Lloyd. I mean, that is certainly critically important, and the sort of the impact we can drive with the existing genetics data and genomics data is very, very powerful. So the goal here is from the sort of collection of cohorts from all over the world, in terms of the surveys we've conducted and those who have signed up. We have about 2.5 million people. The vast majority of them have GWAS data, about half of them have sequencing data, and we are going to generate topologenic risk scores and sort of inform scores also with additional phenotypic risk factors and family history, and those will be utilized to drive sort of the highest risk individuals that we will subsequently want to invest in, because the majority of these cohorts, they have actually ability to recontact these individuals. So if the most informative individuals in the age range, say, of 50 to 65 that have large longitudinal data of collection over that time period are available, so we can do imaging studies on them. If they don't exist, we can do digital biomarker monitoring and assessment. That becomes extremely powerful. It also, when we have such a large scale cohort, we can obviously identify rare variants that have never ever been able to surface because they are too rare for the sample sizes that have been sort of analyzed before. So that allows us for, so another dimension, if you will, of identifying potential new pathways, new gene networks where we have molecular sort of PCSK9-like explanations potentially for the validity of those rare variants. So that becomes, extremely valuable and powerful as a sort of, baseline assessment, and to further invest in those genetically and genomically informed individuals that we can drive.
Whilst an effort like this can be informative for any metric that's measured, I feel the speed to action from genomic insights is likely to be the fastest. Mr. Geek out in the epidemiology, some of the reverse association biases are minimized. With a globally diverse cohort like this, we may discover really interesting, really informative variants that don't exist in populations of European continental ancestry. So I feel like the genomic accelerator of this initiative is going to be key. That links nicely to the next topic I wanted to get into, which is around precision medicines and how do we actually make medicines from an effort like this. So my personal career trajectory, I focused on cardiometabolic diseases to begin with, and then a little bit of oncology, and now Alzheimer's disease. And when I look back with considerable envy on oncology and cardiovascular disease, and I wonder why on earth can we not do what those guys are doing? So specifically in oncology, there the game really has been changed to move away from traditional medicines focusing on organ-based diagnosis of disease, breast cancer, prostate cancer, and instead focusing on the molecular drivers of the disease, regardless of organ, in a very specific, kind of molecularly defined precision medicine population kind of way. Why can't we do that? And what would it take to allow Alzheimer's to get there? So Andrea, I'd love to get your thoughts on how can Alzheimer's be more like oncology, and how and when will we start to realize precision medicine?
Yeah, it's a question which goes very much to my heart. As you know, I'm coming from the cancer field. Before I answer your question, I would really like to thank George and Arno, and now, of course, Nadeem as a chairman, and the COI area and members of the Davos Alzheimer Collaborative to really bring this together. I'm proud that this Lausanne dialogue is now in the 7th year. And I think we have really gone from strength to strength, and I think contributed to the Davos Alzheimer Collaborative. I think when we are all in this midst of this COVID-19 crisis, and when I was thinking about it, I felt like, okay, this year 2020 feels like a decade. But I think there might be also something positive, because what it really teaches us is that a global crisis needs a global leadership. The leadership in Alzheimer's disease actually could come from this Davos Alzheimer Collaborative, spearheaded by George and Arno and the World Economic Forum, and of course, highly supported by the members. So congratulations to George to actually action what we have discussed in Lausanne VI. I'm very proud of it. Now, to the question, I think when we speak about precision medicine, and that's where we are going, we have actually learned in cancer to really go from mutation, which led to an organ-specific treatment, to actually a pan-cancer treatment. Now, unfortunately, in Alzheimer's, we cannot biopsy the brain very easily, so we need to go to other technologies such as imaging, such as molecular profiling, and look for not genes, but actually necessarily, but also the proteins, which in fact are at the initiation of the disease. So I think what I really think is critical and linked to this global cohort is really to identify specific targets which allow us to find the right patients, to actually identify the right markers to again look for the phenotype also which we are treating. And I think in analogy to cancer, I would really like to see that we are looking at Alzheimer not as a single target, because it is not a single target disease, but actually look at Alzheimer as a multiple target disease, and which actually goes back again to the cohort, a multiple target disease which we have to understand, and where we have the right biomarkers to select the patients, to select the drug, to select the endpoints, and all of this hopefully can come together with this cohort. I would even go one step further than very much where we were in cancer today, and actually see maybe in the future we should not even think about treating Alzheimer. Maybe we should think about treating tauopathies, alpha-synucleinopathies, TDP-43opathies. So actually taking this as a pan treatment for neurodegenerative diseases. Now, I've been thinking a lot about this cohort and how I can profit from this cohort as somebody very much interested in Alzheimer research. And I think we are even too moderate, because one of the big questions, the open questions right now, is actually how different targets contribute to the disease, to the onset of the disease, as well as to the progression of the disease, how targets actually interrelate and interact and drive the disease progress, and how, in fact, the presence of specific targets can really influence treatment success. So this all points towards a better understanding of the disease, and this again goes back to what this cohort could teach us. So I'm really extremely interested in understanding some of these mechanisms, understanding how targets correlate with biomarkers, how targets correlate with phenotypes, and how, in fact, we can use this phenotype to predict the right drug for the right treatment. So I think when you look into this in a global way, right now, we don't even have the mouse models which allow us what the contribution would be of a certain target. Let's take TDP-43 to, for example, the disease progression. We don't know. So again, I would expect that these cohorts really allow us to link genotype, phenotype, biomarkers, even environmental markers to a certain cohort. So that's my wish, my dream.
Excellent. Thank you, Andrea. And just to echo what you said, I would love it in our lifetime if there was no longer a disease called Alzheimer's. And that might be because we've eradicated it or cured it. It's probably unlikely. Or it might be because we've redefined Alzheimer's into its molecular drivers, just like cancer. And like you were saying, instead of trying to make a drug for Alzheimer's, we'll make a drug for tauopathies and gliopathies, astrocytopathies, whatever the pathophysiological driver of the disease is. And to make a drug like that is a fundamentally different process and a much more kind of defined process than trying to make a drug for a kind of confusing multifactorial kind of cluster of mechanisms, and then try and hope you can find the right patient for it. And also just to add to what you said regarding kind of sources of drug target ideas, I feel very fortunate working in this space in the current era. But even 10 years ago, our toolbox for understanding the human biology of the disease was very sparse. We had to rely much more on mouse biology and then hope insights from mice would translate to humans. And we no longer need to do that. We can study human biology in unprecedented detail, scale, and speed. So we can get insights into the human biology causes of disease, and therefore have much higher confidence that any drug that we make will actually be able to target the right molecular mechanisms.
Fully agree.
So just segueing back to my other analogy of my past life in heart disease. So whereas in cancer, I was very envious of their ability to understand pathogenic subdrivers and realize precision medicine. In cardiovascular disease, I was very jealous of their ability to predict risk of cardiovascular disease. And of course, a primary reason they can do that is the Framingham Heart Study. And Rhoda, it's an absolute pleasure to have you on the panel today. And I wanted to ask you, kind of from your perspective, retrospectively as well as prospectively, and we now have the opportunity to do the Framingham Heart Study for Alzheimer's, but in the 21st century. So what does that look like? What can we do today? What are the tools that we have today that we simply didn't have before? And what opportunities does that present?
Sure. I mean, I'd like to just sort of step back and just remind everybody that when Framingham was actually first initiated in 1948, we actually didn't know what are the factors of the risk factors for heart disease. In fact, the concept of risk factors hadn't even been developed yet. So Framingham actually coined the concept of risk factors in itself in its first publication in 1961. And really sort of laid out. So sort of the pathway. And I think we're kind of at that same stage here with respect to Alzheimer's disease. We have, again, we don't really have a sense of what's causing this disease, not in its entirety, right? So we know lots of people are affected by it. We know lots of people are dying about it, just like we did with heart disease and stroke back in 1948. And we had some thoughts about what those elements might be that are contributing to it, just like they did in Framingham years ago. But there was a certain sort of what I would say leap of faith that had to happen in building Framingham with respect to heart disease that I think we're now sort of pushing forward. We have greater knowledge. We have genetics, that we never had, the whole multi-omics piece that we didn't have before, but we also have lots of challenges that we don't have before. So one of the things I like to point out is that we have a much more mobile population than we ever had. So the idea of that we want to take a Framingham-like approach, which is really to follow people over a long period of time for a disease that's really insidious in onset, and that we have now determined may go back as far back as 10, 20, 30 years. In terms of its etiologies. So that's a challenge today that we have to confront. I think the other thing is that Framingham, for all its strengths, is largely a European population, or European, based population in its ancestry. And we need to do this in a much more inclusive way than we have ever in the past. And I think the other thing fundamentally that I like to embrace is this idea that we're going to go down paths, just like we did Framingham in 1948. We're going to go down paths that we don't actually really know yet. We have some inklings of some pathways, but probably the majority we don't even know. And so what we have to do is think about how do we do a better job of sharing with the broader community so that we can accelerate the insights in a way that hadn't been possible when Framingham was first initiated. And I think that with respect to AD, I think the other thing, an important lesson that came out of Framingham is, I obviously have spent the last 30 years there focused on brain-related disorders and specifically Alzheimer's disease. But Framingham was a heart study. It never was developed with that intent. And it actually, in 1976, which is really the foundation for Framingham's AD studies, when they first administered the neuropsych battery for the very first time, they didn't actually even know why. The NINDS ADRDA criteria for Alzheimer's disease hadn't even been published at this point. And so I think the other challenge is how do we anticipate what we don't know yet? And how do we set the foundation? And I think the other important lesson of Framingham is that sometimes you have to go outside the disease to fully understand the entirety of the disease itself. And we know that AD has a lot of heterogeneity. So I think that the approach that the Davos group is taking, in not just looking at AD-specific cohorts, but going beyond that, and finding other disease of interest and bringing that in will help us much more comprehensively understand AD specifically in the way that we've now been able to repurpose Framingham from the heart to the brain.
Just to use that to segue on to the next point I wanted to bring up, be great to get your thoughts first, Rhoda, and then I'll ask Elias to add to it. And that's around the digitization of medical research and the digital revolution that we have. I feel like our ability to study the human phenome in all its forms, the scale, the pace in real time the tools, again, we have today didn't exist even 5 years ago, let alone 60 years ago. From your perspective, what are the specific opportunities digital technologies provide for trying to better understand disease cause and trajectory?
Yeah, I think that what digital technology allows us to do is obviously take our data collection methods and take them away from the in-person, in-clinic, sporadic methods and bring them into people's natural environments. I think that then we can do it on a much more continuous basis. We know there's a lot of heterogeneity and behavior. And I think, before we've been taking snapshots in time that may or may not be accurate reflections. The other thing is, again, this is an insidious onset disease. And so we're not picking up subtle changes. We've made the point that earlier is very important, and yet the tools, the methods that we've been using don't really allow us to detect that in any level of confidence. I think that there was a lot of discussion about the need that as we move in these new directions, we need to collect information that gives us confidence in what we're collecting. And I think being able to do it on a much more frequent, continuous basis and let us really, capture not just the heterogeneity, but then let the patterns that need to emerge come out of that in order for us to better understand the full complexity of how this disease manifests itself, particularly in the earlier stages. And so I think that we're actually at a point where we have an opportunity for a very significant paradigm shift in rethinking about how we do things and what we think we know, and also how do we explore what we don't know.
And Elias, if I can just ask you to add to that in terms of the incorporation of current technologies, cutting edge technologies into the Cohort initiative, as well as kind of future proofing for new technologies that may emerge. It'd be great to get your perspective on how you're thinking about this.
Yeah, so it's a very good question. I think Rhoda answered part of it. The key really is what you said. We have tools now we didn't have 5 years ago. The ability to aggregate enormous amounts of data and do analytics that we couldn't do before is really going to be essential. Why? Because you're going to accumulate through this cohort enormous amount of new data that are not validated against a particular natural history, and the ability to do it across a very diverse cohort. I think extracting knowledge out of that is going to be a big challenge. This is actually one of our pillars. How do we do data analytics online, real time, for patients, for subjects, so that you can track, in fact, the evolution and appearance of the disease? Now, we know already that digital biomarkers have already shown themselves in the cognition space, psychiatric diseases, as very powerful tools. For example, in bipolar disease, we can actually identify the manic phases and the depressive phases just by the use of a cell phone. The same thing is true with the Alzheimer's disease initiative. With the UK Biobank, they really had voice prints and checking the changes in the voice and the way you type. And they correlated that with the genomes. And they found actually a very high correlation with ApoE4, homozygosity and heterozygosity. ApoE4 being the only well-known predictive biomarkers, but we have no idea how this biomarker is functioning. So imagine a world where we would have ApoE4 as a clue, right? I mean, it's almost like a string out of a ball of very complex relationships and pathways. And then we would find out maybe in certain populations of the world where there is ApoE4 homozygosity, the patients don't develop the disease. So understanding what is protecting patients from Alzheimer's disease is almost as important as to understand what is it that makes people sick and ill with the disease. So we need to understand the drivers of the disease, and we need to understand the protectors of the disease. And we need to do this with biomarkers that are reliable. So new technology in what we call liquid biopsies, very highly sensitive detection of proteomics within the blood, are already showing that you can actually identify what Hakon was saying, the constellation of risk factors that enrich the population towards what we want. But for us, I think we're at the beginning of a journey, I think we need to truly make sure that this heterogeneity is not a nice to have, it's a must have. Because if you didn't have the ability to compare and contrast environments, biomarkers, but to do this analysis coming from the digital methods and the lab methods that we have today, you couldn't do it 5-10 years ago. So that's really what I think is the key here, is to be smart enough, hence the point that you made, that we need multidisciplinarity and we need to find synergy between areas of knowledge that are very complex in themselves. And then at the same time, are needed in a synergistic way to sort of advance the field and do what Rhoda said. In other words, go into the unknown, knowing, to have guiding lights and guiding cords, if you will, that brings us back to the ability to treat the disease.
Nadeem, this is Andrea. Can I ask a question to Elias and Hakon? Short. Hello?
Go ahead. Sure.
Yeah.
Yes, go ahead.
So my question to you is, how is there a role foreseen for genetic populations such as Down syndrome, particular mutations, presenilin mutations, etc., or are you really focusing only on new findings, more heterogeneous populations? The findings, the learning, homogeneous populations, genetic populations might be important for a project.
I can start. I mean, that's a great question, Andrea, and certainly very, very important to address that in parallel. I mean, we have populations of cerebral amyloid angiopathy, tau protein, prion proteins, and other proteins in almost, all over the world. You have the British, dementia, familial dementia, Danish, Icelandic, through the cystatin C amyloid aggregation that leads to dementia in the 30s and 40s. I mean, these are critically important populations to study also in parallel, because I think that, they have the same underlying process. It's just accelerated at a rate which is, on a log scale compared to an average person. So I think understanding those variations as well as the heterogeneity in diversity populations. I mean, we will never sort of really fully, understand the disease unless we bring those together. But I think that's really, really important and will be a focus as well.
And if I may, on the strategy that we put together, if you recall, I described the sort of the trunk of a large tree, which is the must-have that the general population would have. And then what I call extreme phenotype branches. When extreme phenotypes emerge, this is where we need to really create a subgroup, if you will, around these extreme phenotypes. This is the example of PCSK9. When it was discovered because two families in France had a gain of function mutation, which drove and accelerated the disease process. So I think you're completely right, Andrea. Extreme phenotypes are going to be very critical to understanding the complexity of this disease.
Great, thanks so much, guys. So I'm going to ask one last question that I had, and I'm going to start going through some of the questions that have been coming in, and please keep sending them in. These are great. And the question I had also links to some things people have been asking. It's around trust. So working in the pharmaceutical sector and being interested in health data, I feel like I'm at the kind of epicenter of public and political distrust. No one likes big pharma right now. No one likes people who want your data. So how do we get people to trust us with their data? What would be the reason that people feel confident and comfortable that we're going to use their data ethically, responsibly, safely, securely, and hit that balance between allowing people who need to access the data access and openly share it so that the scientific insights can be accelerated and are not restricted to any kind of subgroup, but at the same time protect the integrity and the quality and the safety of the data. Hakon, if you don't mind, can I ask you to start?
Yeah, thank you, Nadeem. I mean, that is similarly a very, very critical question, because, I mean, you know, we will never go anywhere unless we have that sort of trust. And we try to build that as effectively as we can. We have, for example, we have a charter that we have everyone sign among the cohorts participants. So the IHC Consortium with over 100 cohorts and over 60 million individuals, over half of them have already signed a charter. And there we sort of lay the ground rules in terms of how the all data is de-identified, and sort of, you know, you will never be able to identify an individual in that setting. We have established a portal we reference as an atlas, where we have as secure of a cloud-based system as we can. We have the Gates Venture program involved with us to optimize that as much as we can. And we have ethical and regulatory people on board to sort of drive these decision makings. So we are doing everything we can, but I mean, you know, I assume we can always do better. But I mean, that is obviously the foundation of being able to move forward. That is that type of trust and rigidity and infrastructure which we have tried to establish.
And Andrea, if I can ask you to speak on this topic next. Personally, working in the pharmaceutical industry, I feel like our sector doesn't do a particularly good job in communicating to the scientific and public community what we do with data. There's often this kind of cloak and dagger suspicion that, you know, we want to monetize data and steal it and hoard it and keep it to ourselves. So what can the pharmaceutical sector do to be able to better and more accurately reflect why we're interested in data, how we use it, and why actually it's advantageous for our sector to be involved in initiatives like this?
So it's a very good question. It's a complex question. It's a difficult question. But I think the structure of trust is different when it comes from a clinical trial with a drug, with a potential profit at the end of the day, as opposed to the structure of trust when it comes from a cohort of patients who are participating. For example, I always heard this issue of trust with the minority population, and African Americans don't participate in trials, for example. And a lot of people said it's trust. There's a lack of trust because of the past and so on. What I found when we looked at it in details and we did surveys is that most of the time, what happens is that there's no there's no connection, if you will, between the investigators and the subjects. And when you ask people, I want you to participate, usually you have trust factors that relate to the personal history of the subject here. In many cases, what you find in Alzheimer's disease are individuals who've been affected in their family, a father, a mother, a brother. And so you have what I call privileged spaces of trust. And this is what you need to do. You can't resolve the issue of the general structure of distrust that is emerging against institutions, against vaccines, against the industry, and so on. And I really don't think that you can sort of one size fits all fix the problem of trust. And that's what Hakon is doing by saying, look, let's create a safe harbor here. Let's create a safe space where there is open consent, there is an understanding of what it is that you do as a subject. Look at vaccines. I mean, look at the number of people who volunteered in the context that was a distrustful context. So I really think that this issue of trust is a great issue at the general level. But when it comes to doing things, you will always find trusting subjects under the constraints of an ethical approach, as Hakon is saying, with proper access rules and proper sharing of the information back to the subjects. And when we did that, and Rhoda may know the Jackson Hart study, the All Hat study, this trust or distrust just melted away. And so you can't generalize, in my view. You just have to get it done. And if you don't try, you'll never learn how to create that trust. But trust is not something that you can basically engineer for the whole world to sort of start loving the pharma industry tomorrow. That's not going to happen.
Yeah. And personally, I feel like the biggest way of gaining trust is by delivering impact. I think there's no better motivator and driver than helping people realize that we did this, and as a result, we now have this. I think the Framingham Heart study is a tremendous example, right? That we did this cohort study, now we can better predict and treat heart disease, the world's biggest killer. So I think impact and communicating what we're trying to ultimately do is often the key motivator for people to want to be participant, either in giving us their data or in using the data that we collect. And Andrea, I wanted to get your thoughts specifically from kind of the pharmaceutical industry perspective around this question of trust and the ethical use of data.
Yeah, I think Elias said exactly what I would have said. There's not so much to add. I think you, at the forefront of what you're doing must be the ethics of conducting the best possible clinical trial with the best endpoints, the best centers, the best science, state of the art. And if you have, in fact, this situation, the people will actually cooperate with you because the objective is the same. We want to generate a treatment for an untreatable disease, for a complete unmet medical need. And as you know, I'm working a lot with Down syndrome populations and parents. And I'm amazed. I'm amazed how much they actually want to know, how much they contribute, how much they trust us, actually, that we are doing the best for their children. So I think it's all a matter of communication and actually playing an ethical role, doing the best ethical clinical trial, etc., and making sure that you find your end product potentially secure. So I actually have seen, I must say, a lot of support from many of the patients and the families, because I'm very clear. I mean, the goal of my life is to contribute to a cure for Alzheimer's. So,
just building on that, there's a really interesting resource in the UK called Genomics England, which is a 100,000 person genome sequencing initiative is now being built upon that. And as part of that initiative, they have a sub-panel, which is their participants panel. So they have 20 or 30 people that are participants in their 100,000 cohort that interact with researchers, scientists, to learn how those scientists are going to use their data. And I recently had the chance to present to them to describe how the pharmaceutical industry uses their data. And rather than being challenging, confrontational, skeptical, they were delighted. And the reason for that is they said to a person, the primary reason I donate my data, my samples, my tissues, is not to create a great database or develop algorithms or novel statistical methods. I want new medicines or cures or treatments. And I get it, for that to happen, multiple sectors have to come together. And I think that objective, that ultimately the reason we're doing this is to find better predictors, stratifiers, treatments, cures, is a really important message. Just building on that and beginning to go through the questions that I've been receiving, Hakon, one kind of obvious question that may come to mind is, well, if there's already over 100,000 cohorts, what's missing? What have they not done? And why do you need to do something else? And maybe phrasing that differently, what's different about the way we're thinking about this to the way the previous 100 efforts have been thought of?
I mean, that is a great question. I mean, the diversity component is sort of, what is really missing, and sort of, bringing the information together, meta-analyzing across these cohorts. This has not been done either. So these are sort of, critical components that I think will add value, and identifying sort of, where the missing gaps are or information lies, and be able to gap fill and generate those information for more comprehensive sort of assessment. But I think, the power is going to be behind the size. In terms of, understanding rare variants. I have always been a strong believer that rare variants are much more important than common variants, sort of from the standpoint of linking biology to a phenotype. So I think we will, with sort of, by bringing these cohorts together, getting in the diversity of variants we've never seen or understood before, that is going to be very, very powerful in this setting.
One way I like to think of it is as a traditional large cohort initiative is usually kind of trying to address many, many questions, or almost kind of all possible questions. And then you set up a resource that is as broad as possible. And this is importantly different. This is almost unique, I feel, in its monofocus in trying to deliver solutions for a disease. And the only analogy I can think of is the Framingham Heart Study, like Rhoda alluded to. It was a study designed to inform heart disease, and because it was so unusual, it was then used to inform multiple other things. And I suspect the same may happen here. This is a resource created to inform Alzheimer's disease. And to my knowledge, there is not another effort like this, kind of so focused on answering that question. Now, the resource and the data may end up being informative for many, many things. And you never know what will come in the future. Rhoda, one of my favorite papers over the last year was that remarkable paper you guys did in Framingham with the voice recordings from the interviews with the participants from I don't know how long ago. And that's a great example of data you recorded and not quite knowing what the use of it might be, and decades later is delivering tremendous value. And I suspect that will be the same case here. But in terms of distinguishing this resource from other resources, I'm not aware of any other resource that is trying to develop a specific global cohort to inform new predictors, treatments, stratifiers for Alzheimer's disease. Is that a fair comment, Hakon?
Yeah, that is, I think, absolutely, the key point here, that this has never been done before, and it's an opportunity that is one of a kind. And as I said, we can do this sort of in a very informed way by initially assessing what is available, what exists, and where do we need to put our efforts specifically to make it as comprehensive as possible.
If I may add one more point that I think Hakon mentioned in his introduction, which is essential to understand, and that differentiates this cohort from all the existing ones. And that is what I call the up spiral of knowledge, because there will be feedback loops. And he mentioned something. We want cohorts where the individuals can be called back. That's essential. It's like the Decode Genetics program in Iceland, because questions, as Rhoda said, are not known to us today. However, to address some questions, we'll have to go back. And when something is discovered, let's go back and study that. So there's a feedback loop concept here that is based on a common set of standards of quality control, of quality assurance, acquisition of data that is over time interactively working with the population as it evolves. That concept is fundamental to understanding. This is what to me was a unique thing about Framingham. I mean, this is the grandfathers, the fathers, the sons. You can go back and you can question new hypotheses. You can evaluate new ideas in a constant loop fashion, which at the end of the day accumulates knowledge as opposed to fragmenting knowledge. Very fundamental.
We're now coming to the last 5 minutes. So I wanted to end by asking each of you to give me a kind of a 1 minute summary of what is the impact you're expecting this initiative to have for Alzheimer's patients and their caregivers. And let's say 5 years from now, when we come back to Lausanne 12, what will be the new thing we know because of this cohort that we don't know today? Andrea, do you want to go first?
Unmute.
You might be on mute.
Pardon, yeah. If I can have my pyramid, please. I made a slide, actually. I thought to summarize the opportunities, but in fact also I think, far-reaching results we can potentially expect from this global cohort. For me, actually, when you look at the basis, the fundament of this cohort, you can really think it's the Google. I called it the Google of a disease, which allows to stratify the patients according to the genotype, the phenotype, the proteinopathies, etc., really the state of the art. And this, of course, now I'm thinking about how I, in fact, would profit from this great resource. is in fact that it allows you to really choose patients basically off a shelf and focus on disease-relevant biomarkers. So this is, of course, an enormous time saver. And then you could actually come to a master clinical trial protocol, which, as we all learned, will really accelerate clinical development. And then going further up my pyramid, I think we can dream about a biomarker-based clinical development, and finally potentially lead to a situation where we have one pivotal trial allowing the approval by the regulatory agencies, and with, in fact, supported by real-world evidence. So when you look at this, I think summarizing it is a great potential what this research, this resource can actually deliver to everybody, in particular to the patients.
And just to build on that, similarly, whilst currently I aspire to make precision medicines, even if we did, we don't know what we would measure in people to be able to match the right medicine to the right patient, because those biomarkers don't exist. And even if we had the right biomarkers, the ability to practically recruit those people into a trial in a targeted and efficient way, it doesn't exist. So I think there's some very practical deliverables this resource might have for drug discovery and development. Rhoda, what are your expectations from this resource?
Yeah, I think that there's sort of a couple things. So on the one hand, we're now talking a lot about digital. And I think pragmatically, because I take sort of Andy and Klaus's comments from the last session to heart, is we have to do some pragmatic incremental work, which will require a lot of investment in terms of really identifying digital biomarkers in the same way that we have invested in understanding blood-based biomarkers as well as neuroimaging biomarkers. So I think that the role that digital has to play is going to be pretty significant going forward. But what I would ask as sort of a bold statement is for us to not consider digital as just a better version of what we're already doing. I think that there's the opportunity to go well beyond what we currently can actually even imagine. And I think in parallel, what I would like to see, and certainly over the course of the next 3 to 5 years, is a bold embrace of this unknown and to really take the risk in some of the directions that we're going to take and not tie them completely to precedent. I think that in order to find what we don't know, we have to actually uncouple from what we do know. And so I think that we have to do both in parallel if we're really going to fully realize, because I think that the digital era is going to completely shift the way in which we do things right now. And I think 5 years from now, we'll be doing things dramatically differently.
I couldn't agree more. And digital technologies, I think, will not only be a more convenient way to phenotype people, but in themselves, maybe a way to diagnose and maybe even treat people. So I think the whole paradigm may be fundamentally shifted by digital technologies. Hakon?
Yeah, so the genes, they obviously harbor all the information about who we are and regulate all biological processes in health and disease. And so having all of these, biomarker data, phenotypic data, digital and imaging data in hundreds of thousands, if not millions of individuals of a diverse sort of patient populations, bringing all of that together. There's no question it's going to open up new biology and link sort of phenotype to a genotype in such a way that we've never seen or understood before. And I think this is going to be the driver for new opportunities in terms of understanding the biological processes in Alzheimer's and dementia and finding and figuring ways of intervening with precision-based approaches.
Excellent. And Elias, I'll ask you to give us your closing remarks.
Well, I think a lot has been said. I mean, if you ask me 5 years from now what I would like to see, and I'd like the world to consider this cohort the cohort of reference for Alzheimer's disease. That when we want to validate predictors of the disease, that this cohort plays a role. When we want to discover new pathways of the disease, this cohort plays a role. That at the end of the day, we'll call this cohort the way we call the Framingham cohort today, the equivalent of framing him for Alzheimer is what I'm dreaming of in 5 years from now.
Terrific. Well, thank you, all of you, for a really interesting discussion, and thank you for allowing me to moderate and hang out with you guys for the last hour. It was a real privilege. And the final thing I'll close with, and I pass back to George. It makes me realize what a privileged position I am in, and we are in, as medicine-making scientists in the current era. The things that we can do, the technologies, the scale, the speed, the quantity, the quality, was sci-fi, literally 5, 10, certainly 15, 20 years ago. It was unimaginable for us to be able to do the things that we can do today. And that gives us real unprecedented hope that with these tools, we should be able to deliver transformative breakthroughs for patients and caregivers. And with that, I'll pass back to George.